Hello, everybody. I'm very happy to have the opportunity to introduce this symposium that AMRIT is sponsoring today. As you can see, the title of the topic is Drug Development, Where Do Our Medicines Come From? My name is Mark Summeray. I'm the Chief Medical Officer at AMRIT Pharmaceuticals. By background, I'm a physician trained in the UK, but I've spent the last just over 20 years of my career developing new drugs for treatment of patients with rare diseases. I'm delighted to be able to also introduce Professor Charlie Craddock, who's joining me as a member of the faculty. Professor Craddock is an academic director for the Centre for Clinical Haematology at Queen Elizabeth Hospital in Birmingham and Professor of Hemato-Oncology at the University of Birmingham. So this is the program that we'll be going through today. Professor Craddock will be presenting a discussion of the approach to evidence-based medicine and how we use and generate evidence in the context of clinical trials to support new product development. And then following his presentation, I will give a presentation that specifically focuses on rare diseases and how we go about drug development in that context and some of the challenges that we have that are specific to rare diseases. And then finally, at the end of the symposium, we will have an opportunity for questions and answers, and I will summarize the proceedings. Thank you very much.
I'd like to thank the organisers of this symposium for the invitation to talk about the exciting opportunities and challenges that present themselves now for us to work together as clinicians and patients to develop and deliver new treatments. These are my disclosures. So this is really a time of almost unprecedented promise for developing new and often transformative therapies to patients with a whole range of diseases. Many of these diseases are long-term, debilitating diseases. Others are commoner diseases such as heart attacks, multiple sclerosis, and others, and it's an area I'm particularly familiar with, are cancers. But what's common across all these diseases now in 2021 is the prospect of transforming the natural history of these often terrible diseases by using therapeutic interventions that we couldn't even have dreamt of 10 or 20 years ago. And the reason for this has been the transformative advances in our understanding of science, starting in 1953 with Watson and Crick's original discovery of DNA. And over the next 50 years, we invested trillions of pounds and dollars as a community, tax dollars, tax pounds from people across the world to understand the basic mechanisms by which these diseases developed. And knowing that mechanism, it's then possible, using different scientific techniques, to develop new agents, new molecules, new drugs that potentially target the abnormality in the cells. And so the ability of the biotechnology and the biopharmaceutical sector in the last 10 years to rapidly develop new therapeutic agents presents at last an opportunity to intervene and improve patient outcome. But with this therapeutic tsunami comes a similar responsibility to ensure that our assessment of the activity and safety of these new drugs is as effective as possible. And one of the challenges we're going to talk about is the fact that the infrastructure we have at present for clinical trial delivery to assess these new therapies largely reflects the very limited number of new drugs we had 20 years ago. And so working together with patients and clinicians and drug developers, it's so important that we think about ensuring that the ability that we have to develop the pivotal clinical trials that's going to allow approval of these new drugs is as swift as possible. And that's what we're going to be talking about in the next few minutes. So the first, I think, and best example of the new world we're living in, I personally experienced on the first week of December in 1999, when I was in New Orleans at the American Society of Hematology. And Brian Drucker, a brilliant scientist and clinician, stood up and presented to 6,000 hematologists the remarkable success story of the first really targeted therapy in blood cancer. It was a drug called Gleevec. It was designed on the back of brilliant science, and it had the ability just to knock out leukemia cells and not touch any other cells. And so Brian Drucker described how 35 patients with otherwise untreatable blood cancer who were right in the last stages of their disease took this tablet once a day and Lazarus-like were able to stand, go about their normal days, normal duties, and many of them went into remission. And Brian described to a spellbound audience of clinicians that yes, this was the example of what the future would be, using science to design new drugs to transform patient outcomes. And elegantly, at the end of his talk, he quoted Isaac Newton: "If I have achieved greatness, it is only because I stand on the shoulders of giants." In other words, we need that brilliant science to develop these new drugs. So what we are searching for then is really good clinical teams, really good scientific programs, good clinical trials units. But we also need to be thinking as a community about how we put in the research nurses in major centres so that every patient can have access to a clinical trial, how we help our gifted clinicians drive these trials, and how we make sure, particularly in rare diseases, that we have a large enough population to allow us to deliver these trials as quickly as possible. So let's think about the start of the journey. Well, I've outlined already, and I've given the example of Brian Drucker. Drug discovery starts by understanding the science of the underlying disease and understanding what patients need. Once you have that profound understanding, working with the biopharmaceutical sector, you can develop new agents that plausibly will be of benefit. We confirm in the lab that indeed these are the right drugs, that they target the right parts of the cell that need to be repaired. And then working together, we assess the safety and activity of these drugs. And it's a long journey. And what I think we're saying today is that we need to be thinking about how we shorten this journey from drug discovery on the left to exploratory development in very early phase trials to show that the drug is safe, to establish the right dose and to show preliminary efficacy, to finally proving, compared with the current standard of care, that the new drug is superior. So we put here that it takes 11 to 15 years. It can sometimes take longer than that. And of course, for patients, there's an urgent need that we shorten that period. So here it is. We think about what part of the cell on the left needs to be targeted. We work to identify new drugs. We check in the lab in what's called in vitro studies that it looks like it does what it should. We check in animals that it works, and then we take it to clinical trials. So when we talk about clinical trials, which is a phrase we use a lot, but what do we mean? Well, it's a careful, methodical, systematic investigation in patients to demonstrate safety and efficacy of a new drug. Clinical trials include a series of tests specifically at looking this in prospective cohorts of patients. They're only conducted when we're confident that the new drug is indeed promising and is likely to be safe, but they're essential if patients are to benefit from new drugs. And again, I want to focus on some of the terminology we use in clinical trials. So we talk about preclinical studies. So that's the study to identify it's a promising drug. Then you may have heard people talk about phase 1 trials. So these are the first studies, sometimes the first in man. And the focus there is that this drug is safe, and also in establishing the proper safe dose to use. And then we talk about phase 1, 2 or phase 2 trials. These are larger trials, between 60 and 200. And these are focused on showing that not only is the drug safe in a larger population, but also it looks promising. And then if we've got a promising safe agent, the definitive way of establishing its value is to take patients with a particular disease and randomize them to the new agents against standard of care and definitively show, compared with a placebo, that there's an improvement. And then finally, once we have started using a new drug, there's very important long-term safety phase 4 data. So these are the considerations we've talked about. And the reason that the primary focus of particularly early phase trials, but of all clinical trials, is safety, comes from the experience that we had in the 1960s with thalidomide. So some of you may not be familiar with this story, but thalidomide was developed as a non-barbiturate sleeping aid. It was approved without any good evidence, and it was widely used until in 1960s there was a blizzard of reports that children born to mothers using thalidomide had often devastating limb deformities. And as a result, thalidomide was banned in 1962. But more importantly than that, it made the whole drug development community wake up to the fact that we can't ever let an example such as thalidomide happen again, and meticulous safety assessment is key in drug development. One of the best examples about how patients and clinicians can work together to rapidly deliver vital and practice-changing clinical trials came from the suite of recovery trials delivered by UK clinicians during the recent COVID-19 pandemic. We owe a great debt to 2 Oxford clinicians who, right at the start of the COVID-19 pandemic, recognized that if we were to develop effective and safe new treatments for severe COVID-19 infection, we needed very rapid delivery of clinical trials. So quite remarkably, and this shows how it can be done, 9 days after developing, writing up, and approving the first clinical trial in the recovery suite, the first patient was enrolled. The key to the success of the recovery studies was not just the speed of recruitment, but it was that the trials that were developed in concert with clinicians and patients, asking really fundamental questions about what we needed to do, what particular areas of COVID-19 infection needed to be addressed with new therapies. And once those trials had been formulated, as I've said, recruiting quickly. So, the recovery trial portfolio was the first in the world to secure approval of a therapeutic intervention, dexamethasone, proven to be effective in a randomized trial. It also secured approval of tocilizumab, but importantly, it also demonstrated that a number of other interventions, such as aspirin, shouldn't be used routinely because they didn't work. So this was a great success story, and it was a success story because trials opened quickly, they recruited rapidly, they were open in every hospital in the UK. The trials that were delivered were randomized studies, delivered unequivocally important data about safety and efficacy. And finally, they were simple. So when we are thinking about the design of a clinical trial, we have to think about what is it that we want to do for patients. We have to think about how do we measure safety and efficacy? What does that look like? How many patients do we need to get meaningful answers? And how quickly can we recruit? So we often talk about the primary endpoint. This is the key measure for a clinical trial in terms of activity that might be used by the regulatory bodies to approve it. We also measure other measures of activity, secondary endpoints, and sometimes surrogate endpoints. So a good example in rare diseases is sotorasib, a new type of a cancer drug called a KRAS inhibitor that was developed because there was scientific evidence that KRAS was a protein that was involved in lung cancer, that this new drug was able to inhibit this protein, and because it was a rare cancer, clinicians were able to design a clinical trial that allowed them to rapidly answer the question about activity on a relatively small population of 124 patients. And this drug pivotally extended survival for 13 months. So the COVID-19 vaccine went through very similar process of rapid assessment in large cohorts of patients. So whilst clinical trials really are the gold standard for understanding a drug and proving that it works, it's very difficult to deliver them in old people, certainly quite challenging in children or people who may not be representative of the whole population. And so for this reason, something we call real-world data, this is evidence looking at the whole of the population outside a clinical trial, is also an essential adjunct to developing new agents. So the challenge, of course, is being able to pull together all this clinical data and to deliver the trial as quickly as possible. And we have to be thoughtful, of course, that these studies are expensive to deliver. So anything that we can do, working with patients to ensure recruitment is rapid, that assessment of the trial is as speedy as possible, is important. So what does this mean for patients? So I think it means, first of all, that patients have a vital role in helping design clinical trials, in helping write patient information sheets, and in helping us encourage recruitment to high quality studies that patients as well as clinicians have approved. So in conclusion, this is the time for the development of new transformative drugs. We have opportunities now we could only have dreamt of 20 years ago. But we must work together to drive the delivery of pivotal clinical trials as quickly as possible. At the moment, drug development is a long process and costly. We need to think about how do we close trials out as quickly as possible. And it's our duty to patients to ensure that clinical trials are as efficient as possible, that we use new models, new methods for recruitment, so that patients see these new drugs as swiftly as possible. So working as a team, clinicians, drug developers, but most importantly, patients, there couldn't be a more exciting time for drug development. And thank you for your attention.
So thank you very much. Before I get into the detail of my presentation, I'd just like to deal briefly with conflicts of interest. As I mentioned earlier, I'm the Chief Medical Officer at Amryt Pharmaceuticals, and obviously in that capacity, an employee and a stock option holder. So I'll start with an executive summary or an overview of the topics I'm going to go through. First of all, just a general discussion of rare diseases and the characteristics of rare diseases and the challenges that those characteristics present for developing new drug treatments. And building on that, considerations that we must take into account when designing clinical trials that will provide evidence that those new treatments may be beneficial in patients with rare diseases. And I'll give some examples in particular that are relevant to EB, or epidermolysis bullosa. Then I'll talk through what evidence of benefit means, how we design trials to provide data that shows that these new treatments are actually beneficial to patients in a way that's meaningful, and how those data are appropriately communicated to payers who are responsible for making decisions about whether or not to reimburse the costs of new drug treatments for rare diseases. And finally, I will discuss how important it is that we all collaborate together to ensure that we develop new treatments for rare diseases in a successful way. So first, I think there's some very good news, actually, because over the last decade, the pharmaceutical industry has focused much more on development of new treatments for rare disease. And there have been some very notable successes, in particular in the area of enzyme replacement therapy, for example, for diseases like Fabry's disease, as well as quite recently, new drug treatments for spinal muscular atrophy. And these successes, of course, encourage companies to invest further. So why have we been successful? I think it's a combination of understanding where to focus in terms of the causes of these diseases that we're treating, as well as a favorable environment that the regulators like the FDA and the European Medicines Agency have created by providing incentives for companies to invest in the development of drugs for rare diseases. And in addition, an improving reimbursement landscape. So, well, the interesting thing about rare diseases is actually they're not that rare. In principle, if you look at any individual rare disease, by definition, it is rare. In the EU, that definition is communicated as a incidence of 5 per 10,000 population, or affecting less than 200,000 people in the United States. But in fact, if you combine all these diseases together, you find that 1 in 17 people are affected by a rare disease. So in fact, it is not actually very rare at all to encounter a patient with a rare disease. And the vast majority of those are not treatable. So there are no drug treatments or other treatments available. Most rare diseases have a strong genetic component to them. They're usually chronic, and they may be life-threatening, and they're certainly life-altering. And despite, as I mentioned, the fact that there's more incentives, such as orphan drug designation, that regulators have put in place to encourage development, 95% of these diseases do not have an approved treatment. So there are particular challenges in designing clinical studies for conditions that are very rare. First of all, we don't really understand the disease often, so the natural history is poorly understood. There's also obviously few patients to participate in clinical trials, and often those patients have a lot of heterogeneity, which just means that they're very different from each other. So even though we may have a group of patients that all suffer from the same rare disease, that doesn't mean that the individual characteristics of the disease are the same in each patient. And because rare diseases are infrequently encountered by physicians, they're often misdiagnosed or not diagnosed at all for a long time, which means they present at a very advanced stage. And all of these issues present challenges that you can see on the right side of the slide for designing clinical studies, for choosing endpoints, for finding patients and enrolling them in clinical trials, and for getting patients into clinical trials at a time when an intervention can actually make a difference and the disease hasn't progressed too far. So designing clinical trials means choosing appropriate endpoints. And we have made progress over the years in finding novel ways of designing studies such that those endpoints can be used in ways that make use of sophisticated statistical techniques that maybe allow us to draw conclusions based on small numbers of patients. We also have limited or often no standard of care treatment options to compare our new treatments against. So we have to decide, how do we compare this new treatment? How do we know that it's actually valuable? What should we compare it against? And often in rare disease trials, we're comparing against placebo. And of course, we have to manage the risk that the medicine that we're developing will ultimately not be approved. And if it is approved, it won't be reimbursed. So this is something that has been a deterrent in the past for the pharmaceutical industry in terms of investing in development of drugs for these kinds of conditions. This shows you the probability of success in rare disease drug development. And actually what I want to emphasize here is that compared with more common diseases, actually the chances of approval ultimately are better in rare diseases. And that's often because we understand, once we've studied it, the fundamental problem that is at the center of the disease, and then we know how to target it. Nevertheless, though, you can see that there's quite a low success rate, particularly earlier on in development. So if we move now to epidermolysis bullosa and use this as an example, we can see that there's various ways of thinking about how we might address the problem. We understand that it's a genetic condition, so we can focus on the gene problems, the mutations or the changes in the gene that code for proteins that may be not functioning or may be completely missing. Or we might choose to replace the protein instead of addressing the gene. Or we might choose to correct the problem in the cells that are carrying the gene and then administer those cells as a cell therapy. And finally, if we understand the pathways involved that result from those changes in the genetic information in the cells, we can sometimes target the pathways with new drugs, and we call those drug therapy. Of course, not all patients with EB are similar to each other. In fact, every patient is by definition unique, and we have to be very careful to ensure that we choose the right drug, the right treatment for that particular patient. Just because, as I mentioned earlier, a patient may suffer from EB, for example, it's important that we understand which subtype, which underlying genetic mutation is causing the problem, and therefore pick the right treatment to go with that patient's particular problem. Guidelines are extremely important in determining how rare diseases should be treated. You can see an example shown here with respect to best practice for managing skin and wound care in epidermolysis bullosa. And guidelines come from a discussion between physicians who are all experienced, or healthcare professionals and nurses, in managing patients with the disease, and also looking at the evidence that's being generated by doing clinical trials and published in the literature. So what we see is that guidelines are developed more and more over time as more and more evidence becomes available. And what we're also seeing is that the perspectives of patients are becoming more understood and incorporated into future guidelines, and that's clearly a good thing. So engaging the patient perspective to ensure that clinical studies are designed the right way is extremely important. The paper that you see shown on the left of the slide was based on a survey that was conducted to get input from patients about what motivates them and what actually deters them from participating in clinical studies. Getting this kind of information ensures that studies are designed in a way that optimizes the chances of patients being willing to participate and also remaining in a clinical study and not withdrawing because they find it too burdensome to continue. As part of designing clinical studies, of course, and I've mentioned this earlier, we have to figure out what to measure that reflects benefit for patients, as well as what to measure that reflects the safety of a new treatment. And you can see some examples shown on the slide. EB, of course, one tends to think of endpoints that assess wound healing for obvious reasons. But there are all sorts of different kinds of endpoints that are targeting the extent to which wounds heal in terms of how quickly, and also other symptoms that are related to the presence of wounds on the skin, like pain and itch, and those symptoms impact on quality of life. And of course, there are other parameters that are important to determine, in addition, which reflect the safety of a product. And if it's, for example, one that might be applied to the skin, ensuring that it isn't being absorbed through the skin or through the wounds of the skin, and presenting an unintended safety concern that way. This is an example of a typical clinical trial. And you can see that on the left, patients are screened for their eligibility to enroll and then randomized in a double-blind fashion, which means that they're allocated to the new treatment or perhaps placebo or standard of care, and they don't know whether they have placebo or whether they have the new treatment. And neither does the physician or the healthcare professional or the staff that are involved in running the trial. That's what double-blind means. It's important because it eliminates the possibility of bias in the way that the results of the study are evaluated. And then finally, on the right side, you can see patients, once they finish the randomized treatment, they're often offered the option or the opportunity to continue in the study in what's called an open label phase, where everybody is given the new treatment and followed for a long time, primarily to assess safety. Working with regulatory agencies is something I mentioned earlier, and this is a good example of how doing so can result in improvement in the understanding of regulatory agencies and also the guidance that those agencies provide to pharmaceutical companies for developing new drugs. Here you can see an old guidance on the left that really wasn't at all suited for EB, and on the right side, a specific EB guidance that the FDA developed after meeting with the patient association, DEBRA, as well as patients and their carers, physicians, and industry. And as a result of this guidance, the FDA dealt with very important issues that need to be addressed in clinical trial design, such as the selection of the appropriate patient population, what endpoints that the agency considers to be meaningful in terms of reflecting benefit, ways of measuring those endpoints, and also understanding that not all evaluations are feasible in studies of rare diseases like EB, because patients may have difficulty traveling or the procedures may be painful and not tolerable. So there are additional considerations that can be included in the design of clinical trials for rare diseases that address how difficult it can be to conduct these studies, including the use of historical data from previous studies that allow you to compare a new treatment with the outcomes that were observed in patients who were previously treated in the past. And using statistical methods like modeling disease progression based on that historical data, and other innovative study design elements, such as using composite endpoints or extrapolating from adults to children using appropriate statistical methodology. Of course, study design ultimately reflects a collaboration between regulators that have certain requirements, as well as physicians and patients. And when you get input from all of those constituents, then you're able to build the best clinical trial design that incorporates the most appropriate list of endpoints that will generate the most meaningful data that satisfies all of the stakeholders. Of course, it's also important to bear in mind that when you're generating data on a rare disease and a new treatment, you have to consider whether or not this new treatment may deliver an economic benefit in terms of avoidance of costs. This is particularly important when thinking about payers and their willingness to reimburse the costs of a new treatment. And you can see here on the slide that there are multiple different sources of costs in the management of patients with a rare disease. And they can be extremely expensive when you add them all up together. And when you break them down into their components, you can think about how a new treatment may in fact be able to deliver cost savings in addition to those other clinical benefits that I mentioned just previously. So finally, clinical studies in rare diseases have many positives, which I've touched on as I've gone through the presentation. Orphan drug designation is one of the incentives that regulators have put in place to speed up the drug approval process. There's also the possibility of allowing early access to new treatments on a compassionate basis, even before a drug is approved. And there are mechanisms in place that the regulators allow patients to be able to do that. Patient advisory groups and patients are engaged in this process, and more so actually as time is going on. And their involvement has led to improvements in understanding and incorporation of endpoints in trials that reflect meaningful changes for patients in terms of the impact of a new treatment on a disease. And rare disease is now very much in the focus and on the radar for country regulators to devote more attention and more resource to encourage development of new treatments for patients with those conditions. So as I mentioned right at the beginning, I want to end on the topic of collaboration. Rare disease drug development is about partnership. And you can't design clinical studies and you can't convince regulators based on the data you generate from those clinical studies to approve products or payers to pay for them unless you incorporate the perspectives of all of these stakeholders into the way that you incorporate or the way that you execute your clinical trials and generate data that's meaningful to all of these stakeholders. So the establishment of a close collaboration and communication between industry and regulators and patient groups is absolutely key to streamlining the process of drug development. Thank you. So I'd like to end by thanking DEBRA International for the opportunity for AMRIT to sponsor this symposium, and also to thank Professor Craddock for his excellent presentation. Hopefully, through the symposium, we will have provided you some useful background on the considerations for clinical trial execution and new development of new treatments. And in particular, of course, try to highlight how things are different when you're developing new treatments for rare diseases like EB. And now we will move to a question and answer session.